Assalamu alaikum. In this video, we are going to discuss digestion of carbohydrates and enzymes involved in digestion of carbohydrates. What are carbohydrates? Carbohydrates are monosaccharides, oligosaccharides, polysaccharides. For example, glucose, fructose, mannose, galactose, starch, glycogen, etc. What is digestion of carbohydrates and why there is need of digestion of carbohydrates? Digestion of carbohydrates is to convert complex carbohydrates into simpler ones. And why there is a need to convert complex carbohydrates into simpler ones? Because our body can only use simpler carbohydrates. Only simple carbohydrates can be absorbed by intestine and can be utilized by our body as source of energy. Complex carbohydrates are polysaccharides and oligosaccharides, whereas simple carbohydrates are monosaccharides. There are two major sites of digestion of carbohydrates. These sites include mouth and small intestine. In mouth, we have enzyme salivary amylase. Salivary amylase hydrolyze alpha 1,4 bond in starch and converts starch into amylose and amylopectin. Amylopectin is branch structure and it also contains alpha 1,6 linkage. Alpha 1,6 linkage can't be hydrolyzed by amylase. Alpha 1,6 linkage also present in glycogen, which can't be hydrolyzed by salivary amylase. So the end product of salivary amylase is amylose, amylopectin, and in addition to these two, we have other carbohydrates which can't be hydrolyzed by salivary amylase. For example, glycogen, which have alpha 1,6 linkage, cellulose, which has beta 1,4 linkage, and disaccharides, which can't be hydrolyzed by salivary amylase. They are hydrolyzed by disaccharidases, which are present in intestine. We further discuss it. They all move along GIT tube and enter the stomach. In stomach, no digestion of carbohydrates occur because in stomach we have acidic medium. Salivary amylase become inactive in acidic medium, so in stomach no digestion of carbohydrates occur. Now all these move into small intestine. Small intestine, we have two types of enzymes, pancreatic amylase and brush border enzymes. First, we talk about pancreatic amylase. Pancreatic amylase enters small intestine through pancreatic duct. It hydrolyzes starch into maltose. In addition to pancreatic amylase, pancreatic secretion also contains bicarbonate ions, which neutralize the acidic content of food coming from stomach. Next, we have brush border enzymes. They are also called mucosal cell membrane enzymes of enterocytes. Enterocytes are cells of intestine. The mucosal cell membrane of these enterocytes, we have disaccharidases, also known as brush border enzymes. Disaccharidases act on disaccharides and convert them into simpler carbohydrates, which are then absorbed by our intestine and can be utilized by our body as energy. Now we make table of disaccharides to understand them clearly. Here we write disaccharide. Here we write bond hydrolyzed by disaccharidase. Next, disaccharide on which it acts. Here we write the breakdown product of that particular disaccharide. So the first disaccharide we are going to discuss is sucrase. It hydrolyzes alpha 1,2 linkage. It acts on, of course, as the name indicates, it acts on sucrose and convert it into glucose and fructose. Lactase. It hydrolyzes beta 1,4 linkage and as the name indicates, it acts on lactose and convert it into glucose and galactose. Next disaccharide is maltase. It hydrolyzes alpha 1 4 linkage. It acts on maltose and convert it into 2 glucose monomers. Right? Next we have isomaltase. It hydrolyzes alpha 1 6 linkage. It acts on isomaltose and similarly as maltose it converts into 2 glucose monomers. Last but not the least, we have trihalase, which hydrolyze alpha 1 1 linkage. Act on trihalose, form 2 glucose monomers. This is all about digestion of carbohydrates. If you have any question, you are free to ask. Thank you.